Look at your hand. Flex your fingers. Feel the heat of your own skin. Right now, trillions of microscopic engines are burning fuel inside you, coordinating across vast biological distances, running the impossible machinery of your consciousness. But here is the secret that biology textbooks often whisper, but rarely shout: the cellular architecture that makes you possible, that makes every animal, plant, and fungus possible, looks like it was invented exactly once in the 4.5 billion year history of Earth. Why? Picture the Earth when it's still learning how to be alive. Dark, iron-rich water. A heavy orange sky choked with methane. Heat shimmering above an ocean that looks like an oil slick. And beneath the surface, right now, in this ancient past, hydrothermal plumes rise like underwater thunderstorms, belching heavy metals and raw chemistry into the crushing black depths. Life is already everywhere down here. But it's all small. Fast. Clever in the way a match flame is clever. For 2 billion years, life was just single rooms. Bacteria and archaea. Capable of incredible chemistry, but fundamentally trapped at the microscopic scale. And then, against all thermodynamic odds, a singularity happened. Two competing, alien lifeforms merged. They didn't digest each other. They didn't destroy each other. They formed a locked-in, one-way treaty that rewrote the rules of the planet. The mystery we are investigating today is brutal and simple. Every thought you've ever had depends on a kind of complex cell that seems to have been a singular, freak accident. Why did complex cells happen once, and then never, ever again? Come with me. Not into a dusty classroom, onto a deep-sea sampling boat. The deck is slick with freezing salt. The air tastes like diesel and iron. We are lowering a crude titanium sampler into the modern abyss to hunt for the ghost of the Archean Ocean. Because we are hunting for a missing event. Not just in ancient rocks, but in the deep history written inside your own DNA. You are built from eukaryotic cells. Cells with massive internal rooms. Cells with a protected nucleus vault. Cells with an active skeleton, motorized conveyor belts, gated security checkpoints, and synchronized schedules. Eukaryotes are skyscrapers compared to the single-room mud huts of bacteria and archaea. If that skyscraper design is so powerful, so explosively successful that it conquered the planet, why isn't it the biological default? Why does complexity look like a lottery ticket that was only drawn once? Here's the ironclad rule of the science audit, the standard that keeps origin stories from becoming comforting bedtime stories. Any scientific claim that tries to explain the origin of complex life must leave receipts. Receipts we can measure today, inside genomes, inside lipid membranes, within cellular energy budgets, or in plausible early Earth ecology. If an idea can't predict something we could still detect with modern instruments, it's not an explanation. It's just poetry wearing a lab coat. So let's start with the crime scene, fast, clear, and absolutely without hiding behind academic jargon. Life on Earth has three great domains: bacteria, archaea, and eukaryotes. Bacteria and archaea are the prokaryotes. That word literally translates to before nucleus. But I want you to feel the reality of it instead of just memorizing the vocabulary. They are studio apartments. Everything happens in one open, chaotic space. The DNA blueprint is floating there, the metabolic chemistry is sparking there, the ribosomes, the tiny protein factories, are churning there. It is incredibly efficient. It is brutally compact. It replicates at terrifying speeds. But it has a hard limit on its size. Then you have the eukaryotes. Plants, animals, fungi, amoebas. Eukaryotes are the skyscrapers. You don't just have one room, you have specialized floors, hallways, locked doors, specialized corporate departments. You have a nucleus to securely store and manage gigabytes of DNA. You have an endomembrane system, a sprawling postal network to package and ship materials. You have a cytoskeleton, literal microscopic protein beams and cables that dynamically build shape, tear it down, and physically drag cargo across the cell. And most importantly, you have mitochondria to run the massive energy demands of this cellular metropolis. And here is the key player that shows up at the center of this evolutionary crime scene again and again: the mitochondrion. If you only remember one concrete piece of evidence from this entire journey, make it this: mitochondria were once free-living bacteria. I do not mean that metaphorically. I mean that literally. Back in the 1960s, a biologist named Lynn Margulis proposed this idea, and she was laughed out of the room. The scientific establishment called her endosymbiotic theory absurd. But she fought back with receipts. Today, we know she was spectacularly right. Mitochondria have their own DNA. It's tiny compared to the massive genome in your nucleus, but it's real, and it's circular, just like a bacterium's. They have double membranes, exactly like a cell that got wrapped up inside another cell's membrane. Their internal ribosomes look uniquely bacterial. And when we sequence their genes and compare them across the entire web of life, mitochondria sit deep inside a bacterial family tree, closely related to a ruthless, highly adaptable group known as alpha proteobacteria. Let that reality settle in your mind. You are, in a very real, biological sense, a consortium. A fused civilization of once separate life forms. A Russian nesting doll of ancient microbes. Now, the other crucial fact, quiet, but absolutely devastating to alternative theories, is this: all eukaryotes we know of either currently have mitochondria, or their genomes show undeniable phantom scars proving they used to have them. Even bizarre, microscopic lineages that live deep in the mud, in oxygen-poor environments where classic oxygen-burning mitochondria would be useless or actively dangerous, still carry reduced mitochondria. They carry mitochondrial remnants called hydrogenosomes or mitosomes. Like an abandoned nuclear power plant, sealed off in concrete, but still fundamentally wired into the city's deep architecture. That strongly implies something massive. Mitochondria arrived early. Not as a late-stage accessory. Not as a premium upgrade bolted onto a cell that was already complex. Early. Right at the very root of the entire eukaryotic lineage. The power plant came first, and the skyscraper was built around it. So the leading narrative family, the one most modern evolutionary biologists can live with, goes exactly like this. An archaeal host cell and a bacterial symbiont enter a partnership that becomes strictly permanent. But this was not a clean Disney-fied, swallow and done event. It was much more like a long, messy, agonizingly slow coevolution, full of near breakups, metabolic compromises, and escalating dependencies over millions of years. But the second you accept that story, we immediately hit the bleeding edge of our understanding. Because the moment you stop reciting the textbook and you actually try to imagine the physics of it happening, the microscopic world becomes hostile again. Zoom with me, right now, into the boundary of a single cell. Not as a sterile textbook diagram. See it as a turbulent, storm-battered shoreline. The cell membrane isn't just a line drawn on a page, it's a living border patrol. It is hypervigilant. It mathematically decides what counts as self, and not self. It tightly manages ion gradients, the microscopic voltage differences that literally power life like a charged battery. It controls the influx of food, the venting of toxic waste, and the transmission of information. And then, one day, somewhere in this ancient, boiling sea, a bacterium crosses that heavily armed border, enters the internal sanctum of the archaeal host, and somehow isn't destroyed. Everything becomes possible. And everything becomes incredibly dangerous. Ask yourself, right now, if a foreign entity moved into your house, brought its own agenda, reproduced whenever it felt like it, consumed your hard-earned resources, and potentially poisoned you with its toxic exhaust, what on earth would it take for that situation to become a stable, lifelong marriage rather than a fatal infestation? That question is not philosophy. That is hardcore biology. The core mystery is simple to say and brutal to solve. Why did this specific symbiosis stabilize into a permanent merger, apparently only once, and then go on to reshape the entire biosphere? Look around nature. Symbiosis is common. Corals and algae. Humans and gut flora. Partnerships are absolutely everywhere. Even endosymbiosis, entire organisms living inside the cells of other organisms, exists today in certain insects and single-celled organisms. Yet eukaryotic grade complexity, with mitochondria deeply integrated at its absolute foundation, looks like a singular, bottleneck event. To audit that bottleneck, we have to face three massive paradoxes. Three distinct, "this should not work," moments that scientists fight over today. First, the engulfment problem. Modern eukaryotic cells, like your white blood cells, can easily engulf prey using a process called phagocytosis. They literally dynamically remodel their cytoskeleton, push their membrane outward, wrap it around a target, and pull it inside, like a mouth made of living skin. But here is the massive catch: prokaryotes don't do that. Archaea and bacteria have rigid, pressurized cell walls or lack the internal molecular motors to do this kind of heavy lifting. They don't have the actin cytoskeleton and the complex membrane trafficking machinery that makes engulfment routine for us. So how exactly do you get the bacterium inside the archaean in the first place? Do you need a predator that already has a mouth? Or can you achieve internalization without needing a mouth at all? If you feel the logical tension here, good. This is exactly where origin stories often cheat, by silently smuggling modern, highly evolved abilities into ancient, primitive cells. Imagine we're back on that deep-sea sampling boat, staring at a microscope feed on a flickering high-definition screen. Two tiny cells drift close together, not as a predator hunting prey, but as two desperate, starving neighbors. The ancient ocean is chemically patchy. Free energy sources are not evenly spread out like sunlight on a calm summer day. They are much more like isolated, flickering campfires in a freezing, pitch-black wasteland. And in that desperate darkness, a very powerful kind of biological relationship can form. It's called syntrophy. Syntrophy is strict metabolic teamwork. One organism's toxic waste is exactly the fuel the other organism needs to survive. It's not an act of kindness, it's the unforgiving law of thermodynamics. It's the way two weak engines can link together to do work that neither could possibly survive doing alone. And we don't just have to guess about this. In 2015, scientists dredging a deep hydrothermal vent field in the Arctic Ocean called Loki's Castle discovered fragments of DNA belonging to a completely new group of archaea. They named them the Asgard archaea. These weren't just any microbes. Genetically, they are our closest known living prokaryotic relatives. They possess the rudimentary starter kits for complex cell structures. But the real shock came in 2020. A Japanese microbiologist named Hiroyuki Yamachi spent 12 agonizing years nursing a culture of Asgard archaea in a bioreactor, waiting for them to grow. 12 years. When he finally got them under an electron microscope, he didn't see a standard, round cell. He saw something out of a sci-fi horror film. The Asgard archaean had long, branching, tentacle-like protrusions extending outward, actively tangling and wrapping around other bacteria in its environment to share metabolites. So here is a highly plausible, physically tested path that completely bypasses the need for a magical first bite. Close association first, internalization later. Cells can stick together. Cells can form centrophic chains to share hydrogen or electrons. Cells can slowly grow flexible membranes outward and inward. Tiny membrane bubbles, called vesicles, can pinch off and fuse. If an ancient Asgard-like archaeal host had even crude membrane dynamics, some basic ability to bend and sculpt its boundary and extend those long, branching tentacles, then an alpha proteobacterium could become slowly wrapped, entirely tangled, and eventually enveloped. Not swallowed during an act of violence. Enveloped during an agonizingly slow, intimate metabolic embrace. But even if you manage to get inside the host, Paradox 2 hits the hypothesis like a sledgehammer. The conflict problem. Two distinct genomes living inside one cell boundary is a guaranteed recipe for a biological civil war. Evolution mercilessly rewards replication. A bacterial symbiont that realizes it is suddenly swimming in an all-you-can-eat buffet of host nutrients might just reproduce faster than its host, behaving exactly like an aggressive, terminal cancer. On the flip side, a host that figures out how to digest its internal symbiont might do so the second times get tough, effectively eating its own partner to survive a famine. And there is a darker, chemical threat. The earliest mitochondria-like bacterium, running its high-octane metabolic engine, would have unavoidably produced reactive oxygen species, free radicals, highly unstable, chemically aggressive byproducts of respiration that act like microscopic shrapnel. Sparks violently flying off a badly tuned engine. Sparks that can easily mutate DNA, shred proteins, and burn the cellular house down from the inside. So what miraculously aligns their incentives early on? What specifically prevents the symbiont from turning into a deadly parasite, or prevents the host from treating it like an afternoon snack? If you want to feel the weight of this problem in your bones, picture a walled medieval city that suddenly allows a highly advanced, heavily armed foreign power plant to be constructed inside its walls. The power plant is undeniably useful, yes. It brings light and heat. But it has its own private security forces, its own internal communications, and massive, unstoppable fuel demands. If the city authorities cannot legally and physically control it, the power plant will inevitably control the city. This is precisely where many pop science explanations vaguely wave their hands and say, well, over millions of years, cooperation evolved. And yes, it eventually did. But our audit demands a physical mechanism. Cooperation is an end result, an outcome, not a tool. What physical, molecular tool could have existed early enough to keep this volatile partnership from violently tearing itself apart? And that brings us to paradox 3, the massive, looming shadow that haunts this entire field of biology, the why not many times problem. If this cellular merger is so incredibly beneficial, why don't we see it happening repeatedly in the fossil record or in the modern environment? Earth has had 4 billion years of biological time. Trillions upon trillions of microbial interactions happen in a single drop of seawater every single day. If the evolutionary pathway to a complex eukaryotic cell is relatively easy, then it should be common. If it's common, the tree of life shouldn't just have one branch of complex life, it should be a dense, tangled forest of independent, weird eukaryotic architectures. We should see cells with totally different ways of organizing a nucleus, or with alternative, totally unrelated organelles powering their growth. But that is aggressively not what the genetic evidence suggests. Eukaryotes look entirely like a single, massive lineage tracing back to one common origin point. LECA. The last eukaryotic common ancestor. So either the event is extraordinarily, astronomically rare, or it actually happened multiple times and only one ruthless lineage survived while the others were driven to extinction. Or we are completely missing evidence of other origins because they left zero descendants. And that distinction deeply matters, because it drastically changes how we think about the prevalence of complex life in the entire universe. It shifts the math on whether human-level intelligence is a highly likely endpoint of biological evolution, or a fragile, nearly impossible accident. Now, I am going to intentionally slow down the pace, because this is the exact moment where the origin story usually becomes either heavily mystical or deeply cynical. I want neither. I want the narrow, rigorously testable, humanly comprehensible path straight between them. We need a hypothesis that makes, only once, feel less like divine fate and more like a treacherous mountain pass. A biological route that absolutely exists, but only opens up under a staggeringly rare alignment of terrain, weather, and timing. Millions of hikers reach the base of the mountain. Most freeze. Most turn back. Only one finds the pass. But beyond that pass, the land is so fertile, so incredibly rich, that the descendants of that one hiker completely flood the world. Here is the theoretical proposal I want to rigorously audit with you today. Let's call it the One-Way Ratchet Hypothesis, the precise alignment of bioenergetics, membrane physics, and deep ecological stability. The claim is not that evolution inherently wanted complexity. Evolution has no foresight. The claim is that extreme complexity strictly required a rare, triple biological alignment. First, you need an archaeal host already living precisely at the ragged edge of energy limitation. Think of biological energy limitation like living fiercely paycheck to paycheck, but for cellular chemistry. A prokaryotic cell has a relatively small surface area compared to its internal volume. Because most of its energy-making machinery, the tiny ADP synthase motors, has to be physically embedded in its outer membrane, there is a brutal, mathematical scaling problem. As a cell gets bigger, its volume increases much faster than its surface area. It physically cannot generate enough power to support its own bulk. To put it in human terms, if your entire national economy can only physically fit on the outer walls of the city, the moment the city population grows, the power grid collapses and everyone starves. Second, you need a bacterial partner capable of exporting highly usable energy, or critical metabolites exactly under the host's specific environmental conditions. It is not enough to just say, a bacterium got inside. The bacterial partner has to be the exact kind of biological engine that can run in that specific dark, anaerobic hydrothermal environment and actively produce something the host actually values, like ADP, reducing power, or intermediate metabolites, without immediately poisoning the host with free radicals or demanding so much fuel that the host starves. The partnership absolutely must pay rent on day one. Third, and this is the absolute cutting edge of modern research, a membrane compatibility handoff. This is the most delicate part of the entire audit. Listen closely. Archaea and bacteria have fundamentally different membrane chemistries. They are built from entirely different lipid molecules. It's the biological equivalent of trying to mix oil and water. They do not easily fuse. Furthermore, mitochondria run on extremely delicate electrochemical gradients. They pump protons across a membrane to create a microscopic voltage. It is literal biological electricity. But it is intensely fragile. Membranes easily leak. Lipid curvature matters. If the host and symbiont membranes are chemically mismatched, the proton gradient violently collapses, just like air rapidly leaking out of a punctured tire. And if the gradient collapses, the energy advantage instantly vanishes. Without a massive energy advantage, evolution loses its patience. You don't get a stable partnership. You get instant death. So the One-Way Ratchet Hypothesis proposes this. Once a minimal targeting and control machinery accidentally appeared, once the host cell randomly mutated the ability to legally send its own proteins into the symbiont, physically regulate its division rate, and reliably siphon off its energy output, the biological merger became a locked, one-way ratchet. Reversal became effectively impossible. Not because it was strictly forbidden by the laws of physics, but because the entire cellular system aggressively rewired itself around the partnership. This is known as endosymbiotic gene transfer. The mitochondrion slowly surrendered its DNA to the host's nucleus, essentially backing up its hard drive in the central vault, stripping itself down until it was nothing but a highly optimized engine. It's like moving from two violently competing, separate businesses to a massive, monopolistic corporation with a single shared bank account, a heavily integrated supply chain, and a single legal identity. You physically cannot just separate without collapsing both entities into bankruptcy. Click, click, click. The ratchet locks. And that, in a single, devastating stroke of logic, offers a concrete explanation for why only once. The vast majority of internal pairings fail early due to parasitic conflict. You absolutely need early genetic enforcement. The vast majority fail due to massive membrane incompatibilities and lethal proton gradient collapse. The bioenergetic engine is mathematically delicate. The vast majority of ocean environments simply do not sustain the millions of years of transitional phases required. You need incredibly stable, isolated syntrophic niches where the partnership is immediately beneficial every single day. Only under staggeringly rare conditions do energy stress, ecological stability, and membrane physics magically align long enough for the complex control machinery to evolve. Now, because this is the science audit, we do not get to fall in love with an elegant idea unless it makes hard, distinctive predictions. We need to define exactly what would look completely different if this hypothesis is totally wrong. Prediction 1. If the energetic ratchet is real, the absolute earliest phases of eukaryotic evolution should show massive genetic signatures of intense, ruthless selection on protein-targeting systems tightly coupled to mitochondrial integration. In simpler terms, across all modern eukaryotes, the specific machinery that imports proteins into the mitochondria, and the postal machinery that moves membranes around inside the cell, must be ancient, foundational, and deeply conserved. They cannot be late, optional additions. How do scientists physically test that today? Comparative genomics. Using supercomputers, we reconstruct the exact evolutionary timing of gene acquisitions. We look at which eukaryotic signature genes appear earliest. If the mitochondrial import machinery shows early, deep conservation and locks entirely with mitochondrial genes across every eukaryote on Earth, that strongly supports the ratchet. If mitochondrial integration looks like a late, messy afterthought, or if we find multiple unrelated, weird import systems suggesting multiple independent origins, the hypothesis critically weakens. Prediction 2. The earliest stable biological intermediate should be metabolically interdependent. Syntrophy. That means the first successful partnership wasn't an aggressive hunter eats prey but fails to digest it. It was starving neighbors breathing each other's exhaust. We can probe this indirectly right now by searching the genomes of ancient archaea for specific metabolic pathways that imply deep dependency, extreme hydrogen transfer, ancient sulfur cycling, highly coupled redox loops. Traits that only make sense in deep, dark hydrothermal settings under massive energy limits. Prediction 3. There must be deeply conserved cellular choke points, inflection points where the host permanently gains absolute leverage over the symbiont's replication cycle and energy output. In your cells right now, the nucleus encodes over 90% of the proteins your mitochondria need to function. The mitochondria literally cannot freely decide what they are. They are fed highly regulated components, tightly restricted, and constantly constrained by the host. If the ratchet was strictly essential early on, we should find deep, universal structural features, shared across all eukaryotes, that look entirely like early evolved enforcement mechanisms rather than later evolutionary optimizations. And now for the absolute most exciting part of modern biology, verification that does not rely strictly on inferring the ancient past. We can try to force nature to replay parts of the tape right now in a lab. Method 1. Experimental evolution in synthetic consortia. We build long-term, high-tech co-cultures in bioreactors. We take modern archaeal-like hosts, or engineered bacteria modified to mimic archaeal membrane traits, and explicitly pair them with specific bacterial partners under brutal, energy-stressed, syntrophic conditions. We artificially select for stable, increasingly intimate associations. Then we measure the conflict. Does one partner inevitably over-replicate and kill the host? Do lethal toxins accumulate? Or do control mechanisms spontaneously evolve? If biological stability emerges preferentially under incredibly narrow ecological conditions and heavily depends on the rapid evolution of targeting-like mechanisms, that supercharges the hypothesis. If stable endosymbiosis readily and easily evolves across broad, varying conditions without needing any special control innovations, it totally shatters the only once bottleneck argument. Method 2. A brutal membrane biophysics stress test. Using synthetic lipid vesicles, we physically quantify the compatibility space. Lipid chemical composition. Membrane curvature physics. Proton leakiness. We ask a blunt, unforgiving physical question. Are there specific archaeal membrane chemistries that make housing long-lived internal symbionts energetically catastrophic? If only a microscopic set of membrane properties permits an internal symbiont to survive without massive, lethal energy loss, the one-way ratchet gains massive scientific teeth. If a wide, generic range of membrane types can easily support internal symbionts with little penalty, then the extreme rarity of eukaryotes needs a completely different explanation. Method 3. Ancestral protein resurrection. This sounds like magic, but it's real. We infer the exact genetic sequences of ancient, ancestral versions of key protein import systems. We chemically synthesize them in a lab, and test whether these minimal, primitive systems can successfully localize proteins to proto-mitochondria-like compartments. We are looking for something incredibly subtle, not a fully modern, perfect import machine, but a crude, leaky targeting system. Just enough to start the relentless cycle of control. If even incredibly primitive, messy ancestral systems show partial, viable targeting, that is the first physical click of the ratchet. Now let's return to the narrative story, because cold facts without a lived timeline don't deeply change how we see the world, they just decorate it. We are back in that crushing Archean ocean. The water is violently warm near the vents and deadly, freezing cold just meters away. The complex chemistry changes violently meter by meter. On our high-definition microscope feed, we watch two solitary cells locked in a tight, desperate dance near a black smoker plume. The Asgard-like archaeal host is living on the absolute jagged edge, energy in, energy out, absolutely zero biological savings. The alpha proteobacterium is a high-revving engine, but it is also a massive chemical risk. At first, it merely clings to the outside. Maybe it nestles safely in a small, curved pocket of the host membrane. Maybe it slowly gets partially wrapped by an accidental biological embrace, a tentacle of membrane holding it close. For a long, dangerous moment, it is totally unclear whether this specific interaction is the magnificent beginning of a totally new domain of life, or just the beginning of a very slow, microscopic murder. Here is the precise turning point, the moment the only once stops feeling purely mystical and starts feeling like brutal, inevitable geometry. If the host cell can securely extract even a tiny, reliable benefit, a trickle of extra ADP, a crucially needed metabolite, a novel way to chemically manage severe redox stress, then it can finally afford to grow just a fraction of a millimeter larger. If it grows a little larger, it can begin to experiment with complex internal structures. Primitive folded compartments. Vastly expanded membrane surface area. Tighter, faster regulatory control. If it gains better control, the volatile symbiont becomes slightly less of a wild, dangerous animal and slightly more of an integrated organ. And every single step makes the previous step mathematically harder to undo. That is the biological ratchet. Click, click, click. But why does any of this deep-time microscopic history matter to you, sitting here, right now, in a hypermodern world of glowing screens, concrete cities, and digital calendars? Because your very cells still carry the heavy scars of this ancient, desperate compromise. Every single time you inhale a breath of oxygen, you are explicitly feeding the distant, highly modified descendants of that ancient bacterial engine. Every single time you move a finger, you are literally cashing out ADP currency minted deep inside your own mitochondria. Your life strictly runs on a corporate biological merger. This is not some fluffy, metaphorical, "we are all connected to the universe" poetry. It is a literal, verifiable historical fact. Two vastly different evolutionary lineages were violently stitched together, their endless metabolic arguments eventually settled not by morality, but by ruthless mechanisms, protein import, membrane traffic, deep gene transfer, and absolute host control. And realizing that completely changes how we should fundamentally think about evolution itself. Evolution is not strictly about red and tooth and claw competition. It is also deeply about mergers, uneasy alliances, and aggressively enforced cooperation. Sometimes, the absolute biggest, most dominant leap forward in planetary history isn't developing a sharper tooth or a faster fin. It's inventing a revolutionary new way to share a microscopic power grid. Now, there is a much deeper, almost haunting implication to all of this, one that can easily make the universe feel either overwhelmingly lonely or exquisitely, beautifully precious. If extreme cellular complexity absolutely depends on this astronomically rare alignment of deep energy stress, matching membrane biophysics, and precisely timed genetic ratchets, then it changes everything about how we look at the night sky. Alien microbial worlds may be incredibly common, yet completely, eternally quiet. There could easily be billions of warm, wet exoplanet oceans full of swirling chemistry and fierce microbial adaptation. Yet absolutely zero forests. Zero eyes. Zero complex languages or alien civilizations. Not because the vast universe is inherently hostile to life, but because that one specific, narrow mountain pass of cellular bioenergetics is just mathematically too damn hard to find. But rarity is not meaninglessness. In science, rarity is an open invitation. A harsh constraint sharply focuses our questions. And physical constraints are exactly what we can vigorously test in a lab. So here is a reality model stress test you can run in your own mind right now. Imagine a counterfactual, alternate reality Earth where eukaryogenesis, the birth of complex cells, is physically easy. Where complex, skyscraper cells continuously evolve again and again, just like fins or eyes evolve repeatedly in totally different animals. What would that alternate planet look like? You would logically expect a massive, chaotic explosion of entirely independent complex lineages very early in Earth's history. You'd expect multiple, completely unrelated versions of mitochondria-like organelles running on entirely different chemistries. Multiple fundamentally incompatible protein import systems. Multiple, wildly different deep roots of cellular complexity. The genetic tree of life would look like a dense, chaotic, disconnected thicket. But look at the data. What we actually see in the ground and in the genome looks infinitely more like a single, devastating lightning strike that set a global forest on fire. And now, slow down with me for just a moment of profound awe. Because this is the exact part of the scientific audit that simply does not fit neatly onto a bar graph or a spreadsheet. You are not a single, pure lineage. You are a highly negotiated treaty written in billions of lines of DNA and microscopic folding membranes. Your conscious self sits precariously atop an ancient, alien power plant that slowly, begrudgingly became family. The strict biological boundary between me and not me was never, ever as clean as our human language pretends it is. If that biological reality makes you slightly uncomfortable, good. Intellectual discomfort is the absolute first honest signal that a broken mental model is actively updating. Now, I want to be entirely transparent with you. As a scientific community, we still do not have every single step of this origin story perfectly mapped out. The physical fossil record of soft, squishy microbes is brutally sparse, and microscopic cells simply do not magically preserve their high-stakes drama in solid stone the way dinosaur bones do. But we have something infinitely better and more reliable than ancient fossils: living, breathing evidence. Modern genomes that silently carry the deep, mathematically verifiable scars of ancient, chaotic gene transfers. Sprawling cellular membrane systems that rigidly preserve billions of years of deep biophysical constraints. Cellular energy budgets that still, to this day, fiercely limit what biological structures cells can physically afford to build. And as our modern scientific tools exponentially improve, from single-cell massive parallel sequencing, to atomic resolution cryo-electron tomography, to deep ancestral machine learning reconstruction, to entirely synthetic lab ecology, we can finally push this ancient audit from merely plausible to mathematically precise. Let's return, one final time, to that dark, ancient sea. The biological merger is not a sudden flash of light. It is a fragile, flickering candle in a hurricane that almost goes out 1,000,000 times over millions of years. A desperate partnership that mathematically should have failed in 1,000 different, highly probable ways: lethal digestion, runaway parasitism, chemical toxicity, sudden energetic collapse. Then, somehow, against all thermodynamic odds, it stabilizes. Internal control slowly emerges. Metabolic dependence deepens. The candle finally stops flickering. The cellular light spreads, agonizingly slowly at first, and then with a violent, unstoppable, global momentum. And over 2 billion years later, you are sitting right here, consciously auditing the deep evidence, simply because, in the total darkness, one ancient cell finally made room for another. Here is the final synthesis, clean, sharp, and brutally audited. Eukaryotic complexity is profoundly not just more random genes, plus more geological time. It is a completely new, revolutionary energy infrastructure. Mitochondria directly made massive genomes and hypercomplex gene regulation biologically affordable. But actually getting a functional mitochondrion wasn't just a wildly lucky, accidental engulfment. It intensely required a mathematically narrow alignment of deep ecology, specific membrane physics, and the rapid evolution of early control machinery. Once that specific control machinery clicked firmly into place, the merger became a locked, one-way ratchet. Now, aggressively carry this biological thought experiment into the rest of your day. If your own physical body is fundamentally a treaty between ancient, alien strangers, what other rigid boundaries in nature might actually be completely negotiable? Species? Individuals? Entire global ecosystems? What looks to you like a single, unified organism might just be a heavily stabilized, ancient alliance waiting to be audited. And the next great, massive question, the heavy door we haven't even dared to open yet, is this: if complex biological life is strictly constrained by cellular energy infrastructure, then what exactly would an alien, extraterrestrial version of a mitochondrion look like? Would it absolutely have to be the exact same biochemical solution, or could the universe theoretically build skyscraper cells running on a completely different, unimaginable kind of microscopic power grid? If you want to keep aggressively pushing this audit, click the video right here on the end screen, because it picks up exactly where this deep biological mystery leaves off, and it pushes the audit straight into the next major scientific frontier. And if you want a concrete reason to subscribe to this channel, here it is. Each week, we take a massive scientific claim that sounds permanently settled, and we ruthlessly stress test it until it either shatters or becomes undeniable, always giving you the exact, testable predictions you can track as new evidence actually arrives. But before you go, I have one direct, specific question for you, and it's easy to answer in a single sentence down below. Based on what we just audited, do you personally think only once means complex life is basically mathematically impossible elsewhere? Or do you think it happened millions of times on Earth, and ruthless history just permanently erased the other desperate attempts? Let me know.